Thank you very much, Professor Gazzard, and I think we can all say that was probably the most entertaining talk of the afternoon. And we've been thoroughly schooled, and my job today was to keep us on time, and I think Brian means we can be dismissed for the afternoon. But on the occasion of saying, I'd like to say thank you for inviting me to chair on this session. So my name's Janaki Amin, and as Mark introduced me, I'm a professor at Macquarie University, but more importantly to me, I'm a fellow of the Kirby Institute. I don't think any of us ever leave, and I think that David allowing me to stay on as a collaborator and part of the Kirby family is very important to me. And one of the things that John said to me towards the end of last year is that I'm now a professorial grandchild of the Kirby Institute, so a third generation to come through. And I had the honour of supervising 2 PhD students with David. And I think if you all look around you, the person next to you, sitting behind you or in front of you, is either a PhD student of David's or a student of those students. So the legacy of David will live on well beyond the people in this room and the people who've come to be in this room with us. So one of David's clinical students, as well as his PhD student, is Patrick Mallon, who's here with us today. So Patty trained under David as a clinician and as a researcher. He returns to us from the centre he has set up in Dublin, the HIV Molecular Research Group. So welcome, Patty.
I'm going to set my timer so that I don't get pulled off stage too quickly. So I'd like to thank the organisers. I'm absolutely delighted and honoured to be here to present today on co-morbidities. I'd also like to thank Brian Gazzard for absolutely bombing my presentation before I even start. Anyone recognise this place? This is where I first met David back in 1999, called Landudno. Anyone ever been there? Oh, a few people. I'd never been there since, but I was working in UCL with Ian Weller and expressed an interest in going to Australia for a year, and he suggested I meet this guy called David Cooper, who was over giving a presentation at Landudno. So I drove up from London, took me 4.5 hours. I sat down with David in a very nondescript hotel in a really rainy day, and we had an interview over a beer. And at the end of that interview, I found myself with a job in Australia. Now, one of the things that David said at the time was, you and your wife, you love Sydney, it's beautiful, and the weather is better than here. And that was what dragged me out to Sydney. So I got on the plane with my wife later that year, and David was completely right. Sydney is an absolutely stunning place, a beautiful place to live and to work. The first time I ever set eyes on Bondi, I was absolutely astounded, and I more or less didn't leave the postcode for the next 7.5 years. There were obviously some cultural differences between Northern Hemisphere and Southern Hemisphere countries, which a young Irish guy had to adjust to, but we managed. And I also got the opportunity then to work at St. Vincent's Hospital. So first year clinical, and then afterwards a very enjoyable 6.5 years working within the Centre for Immunology as part of the then NCHECR, which is now Kirby. Now, at that time, it was a very exciting time in HIV. There was a lot of stuff going on. People have said that David was involved in all aspects of therapeutics. The time that I arrived, triple therapy had really just started. Drug toxicity was one of the major issues. And over the 6.5 years that I was there, I got a really nice introduction into what it is to work within a family of very active and committed researchers working in a whole range of areas. And David and the group at Kirby have had significant impacts internationally on every single one of the areas listed here, and have really been instrumental internationally in driving this graph from a red to a green zone. So how do you sum up Coops as a leader? Well, I came across this quote, and I think it reflects what some people were saying yesterday. No man will make a great leader who wants to do it all himself or get all the credit for doing it. And I think there's no truer word said about David. It was reflected in some of the very nice comments that were mentioned yesterday. But to try and tease out what qualities Coops had, I guess we need to go back into the science. And it was through the science that I actually got an insight not only into the Coops as the leader, but also into the family that he created as researchers. So very quickly after joining David, the main toxicity issue at that stage was lipoatrophy, which was reflected in a seminal paper produced by Andrew Carr and David Cooper, who very successfully identified and characterized the condition that was HIV-associated lipodystrophy, which initially was attributed to protease inhibitor therapy. This condition at the time when we were looking after patients really was devastating for the community perspective. It took away individuals' anonymity in terms of their ability to hide their disease. And at the time was considered to be a stigmatizing, but also a very clear threat to the success of an early stage antiretroviral therapy. So I got the opportunity very quickly after joining to work on an NIH grant that Andrew and David have been successfully funded for. And the first project within that was to try and characterize what actually was leading to the development of lipoatrophy, because as Brian said, with everything in science, there's so many different people pitching in. People were saying it was protease inhibitors. Other people were saying it was HIV disease itself. So we set about actually characterizing what happens when you start therapy, and this came up with what was really a sentinel paper at the time that first described this early change in body composition with treatment, where we got an increase in lymph fat and then this very selective progressive loss of lymph fat that continued with treatment, so clearly showing an association with treatment, but interestingly showing an association not with protease inhibitor therapy, but with D4T. And through this research, what I realized was that I was working with a guy who was committed to finding the best treatments for patients and the safest treatments for patients. And he really surrounded himself and engendered qualities in people by leading through example. One of the three characteristics that David had which stayed with him throughout his life were integrity, trust, and loyalty. And I noticed that Tony Kelleher mentioned loyalty earlier on. Loyalty is a two-way street. And David was incredibly loyal to everyone that he worked with, but in return, he expected that loyalty to be reciprocated. And this generated a sense of family and a sense of trust that made the research more productive and more smooth. And he also engendered a pursuit of excellence. It wasn't just enough for David to be able to describe lipoatrophy. What he wanted to be able to do was to drive that research journey through so that he could actually have an end result that would lead to a better clinical outcome. So we turned to science, and this was one of the beauties and one of the things that I was taught by David Cooper that I've managed to bring forward throughout my career, is having the ability of taking a clinical observation, working through the science, and then coming out the other end with a solution. So the science around the time was really focusing around mitochondrial toxicity, and again, the early studies where Australia was really leading the field, in this case our colleagues over in Perth, showed very clear associations with mitochondrial toxicity and use of the thymidine analogues, AZT and D4T. We took that one step further, though, at the CFI, where I moved to after my second year of my PhD, to work with a very talented and loving group of researchers for the next 4 or 5 years, led by a young, talented immunologist who was just back from Oxford. So Tony in the forefront here, I knew that this photo, when I look back on it, was taken in the winter. I'm sure you're wondering how I know it was taken in the winter. Well, there's a direct correlation between the time of the year and the amount of hair on Professor Kelleher's face. So the Australian calendar usually starts off in January, where a very clean-shaven and smart Professor Kelleher goes off on his summer holidays, usually to a bush beach somewhere south of Sydney, disappears for 3 weeks, and then comes back looking like a mountain man. And then what happens as the year progresses is that all of these very caring lab assistants eventually try and convince him that he looks better unshaven, and this usually happens by the time you get into the winter months. But with this lab group, we were able to look at things in a bit more detail. Real-time PCR had just been introduced into the lab, and we were able to start looking at RNA rather than DNA. And what we did was look at the mitochondrial function in a bit more detail. And what we determined by looking in appropriate cases, so in this case we did a randomized prospective study, We took patients without HIV or subjects without HIV, so healthy volunteers, and we actually looked into the fat tissue. So there's a lot of people have contributed. They're now walking around Sydney with small scars on their flanks. Some of them will be in the audience here. Contributed to this data that really conclusively showed that these drugs were associated with very early toxicities at a fat level, and that these were happening even before the mitochondrial DNA in fat was declining. So through these series of experiments working within the Kirby environment, we were able to go through the full process of not only describing a clinical entity, but getting down into the nitty-gritty of the science and conclusively showing a causative association with particular drugs. And the remarkable thing about that is that not only was it demonstrated and repeated internationally in a lot of much larger and more expensive studies, and again with different studies that looked at removal of the drug showing reversibility, but very quickly the science was turned into changes in how we treated patients. And by the time I'd finished my PhD, drugs like D4T and AZT really weren't being routinely used in clinical practice within Sydney and much of the Western world. And this was a 5-year cycle, and I've never seen anything move as quickly in HIV since, where you go from clinical observation to change in practice through science in 5 years. Another attribute that Coops had as a leader was really this fearlessness and a lack of conflict. And I put this up as my disclosure slide for a reason, in that I still have a lot of disclosures with pharma. And it's something that I learned working with David. If you're working in drug toxicity and you're working in comorbidities, you end up, there's a necessity to work alongside pharma. And David knew that from a very early age. When I asked him about conflicts, he said, well, one of the best ways to limit your conflicts of interest is just to be totally conflicted. So David really didn't care who he worked with or where the money was coming from, provided that he was driving the science. And that was a really important concept in terms of that partnership between clinical academics and the pharma industry, which is leading to continued success within the Kirby Institute to this day. The other thing about Coops is that he was trying to find a word to describe it. I came up with this paternalistic leadership style. Now, this is a well-defined leadership style. It's very controversial. His wasn't generated. His was natural. And when you listen to people talk about the Kirby, they talk about a family. And that family sense was there even back in the early 90s when I came to Australia. And I got the opportunity to meet a lot of really remarkable people in and around Sydney through the 7 years that I was here. And these people have remained friends and colleagues ever since. Colleagues that were both clinically based, research based, and colleagues that were both medical and non-medical. But Coops also gave me the opportunity to meet a lot of people outside of the Kirby. And one of the endearing qualities of him as a leader was he may not be a person that gave you direct feedback in terms of how good you were, but he was very direct in terms of telling everybody else how good you were. And I think that's a very strong leadership quality, which I think endures. So it meant that when I got to meet people like Peter Rice, when I got to meet people like Bill Powderly, they had already been introduced to my qualities by David Cooper. And I know that I'm not the only person to have benefited from that. Certainly Alan Winston pictured here. Mark Weinberg's obviously trying to be cool and not trying to make fun of Alan. Alan Winston pictured here certainly has benefited immensely from the same sort of tute leads that I benefited working with Coops. And we got to meet a lot of very interesting people. I'm not sure if any of you recognize this forehead, but he's in the audience. This is Professor Alan Landay, Professor of Immunology at Rush University in Chicago. Not only is he a world-famous immunologist and a fantastic collaborator and friend, but he also holds what I would consider to be the world record for doing the plank in a bar at Croy Conference. So this guy has a core that would really embarrass most of the Bondi Beach Boys. So having enabled me with the skills and the quality to do clinical research, having facilitated the networks that I generated through the research that we've done, in 2007 I managed to get back to Ireland, where I started off my own research group, pretty much starting from scratch under the guidance of Bill Powderly. Even when I went back to Ireland, the loyalty of Coops was still apparent, and that's really where the mentorship comes in. Right up until being awarded my chair just before Christmas, Coops was with me the whole way, providing the support and providing the guidance. And having talked to people over the last couple of days, it's amazing the number of people that have said the same thing. When they had a problem, they went to Coops. When they wanted advice, they went to Coops. And it was that mentorship which was an incredibly strong quality. Since coming back to Dublin, we have a very young HIV population. There is no lipoatrophy. It's an amazing reflection of the advances in science, but we do have an older population. And as Brian said, we're interested in researching that older population. Myself and Alan Winston, amongst others, have set up a very large cohort study. Brian loves it. I must actually just point out that one of the 6 London centres in the Poppy study is Brian's centre at Chelsea and Westminster, but you better not tell him. These cohorts are established to help us understand or observe. As Brian said, there's a lot of controversy. What we need to be able to do is to tease out the controversy and actually figure out and try and understand what's going on. With cardiovascular disease, we've also done a lot of work extending that concept of going back and looking at disease pathogenesis. So the abacavir story that's dogged HIV for the last 10 years. Through the types of research that we started off doing in Sydney, we've now been able to extend that and define within randomized clinical trials specific biological pathways that explain an association that we see clinically. And lastly, just to finish up, there's foresight. And David's foresight is there. The foresight, as Brian alluded to, about trying to understand what it is to live with HIV. What is the relevance of all the inflammation that we see that persists in people living with HIV? Does it matter that we see signals of inflammation that are associated with morbidity and mortality in treated populations? And I think where the field is going, and it sort of ties in nicely with what Brian said, is there are a lot of factors in people living with HIV, some of which are abnormal and some of which are normal. And I would argue that the vast majority of people living with HIV have an essentially normal biology, but there is a small subset who will be less advantaged, and we have markers that would suggest that, but equally there may be a small subset that are much more advantaged. And a lot of these subsets, what we're dealing with is interaction between the host and the pathogen and the immune system. And I think David would be proud at this stage to know that the comorbidities field has really turned full circle back to almost pure immunology. And for an immunologist, I think he would be proud to see the field as it extends over the next 5 to 10 years. In light of the advances that have been made in the field and David's contribution to the field, the Comorbidities and Adverse Drug Reactions Workshop in HIV have recently announced these Cooper scholarships. So for the first time, they're going to enable young researchers to be able to attend these conferences under these prestigious scholarships, and I encourage anyone interested to apply. So lastly, the last quality the Coops had was friendship. And I'm just going to finish with this slide. So I had the pleasure of welcoming Coops and Dory to Dublin a couple of years back, and they say a picture says 1,000 words. And I would like just to finish up by saying to Dory, Lana, and Beck, thank you for the sacrifices that you made that built the environment that enabled Coops' success. And although we miss him and we have our grief is nothing compared to yours, but we do hope that you derive some solace from the fact that the greatness that you have enabled will persist for many years to come. Thank you.